So welcome to video 9 in our course G07 on phylogenomics. And here we're looking at why would we get the wrong tree. So we consider basic things that our sequence alignment approach was maybe incorrect. We may have a sampling error in the species. We may have an incorrect evolutionary model that's being used. We know that the search algorithm, such as neighbor joining, may not identify the most accurate tree, and the optimality criteria is also not guaranteed to get the correct tree either. There's also more complex processes. So if we're looking at it, say, across a group of birds, you may have incomplete lineage sorting. So we have different patterns for different genes that are shown by this black line, the dotted line, and then the dashed line. So the black line we see matches the kind of pattern for the phylogeny. We get these the bifurcations, whereas for the dotted line next, we see here that there's common ancestry here for this species and this species, where this species is more, the blackbird is more divergent at this point here. For locus 3, the dashed line, these two species here and here, emu and grasshopper, woodpecker, have more recent common ancestry compared to the other two, where this wren has common ancestry here at this point here, whereas we see that this the branch leading to the blackbird is actually the most divergent among these four. So that incomplete lineage sorting means that we get what's called gene tree, species tree incongruence. So we can also look at coalescence in terms of this. So think about the ancestry of two particular individuals, and they come together to form a particular ancestor. So think about the present, and we're sampling back into back through time. We're expecting to meet at a particular point. The exact placement on the phylogeny may vary depending on the diversity of the presence of the population that's present. We may have different alleles that are segregating at this point in time, and therefore the perceived fixation times of different alleles may vary and may be estimated. Similarly, if we have a population changes such as an increase or decrease, we can see that for a recent increase, the ancestry will be very shallow. For a population bottleneck, the ancestry may sometimes be quite deep. We also know that this issue of older mutations segregated at the time of speciation or separation may make this more complicated, and that's enabled by this diagram here, where the average trees for ABCD here is shown. But then we might find at different genes, gene 1, gene 2A, and gene 2B, is that we get different patterns where CD are more closely related for gene 2A, and the same for G2B, where the ancestry for A and B may actually be further in time than for C and D. That's most clearly established for the human chimp gorilla patterns, where human and chimps are more closely related on average. But for about 20% of sites, we get other patterns, where, say, chimp and gorilla might be more similar to each other, or human and gorilla might be more similar to each other as well. So that kind of leads to more diverse patterns of diversity due to incomplete lineage sorting. There are ways of getting around this. So there's such thing as the multi-species coalescent model that tries to address this by where the gene trees are fitted to coalescent processes, and that can include gene flow integration. And there's other more complicated, such as hybridization between Siberian tigers and lions, and there's other variations on lions and tigers, ligers, and tyons, and so on, lilligers, and all sorts of variations on this. And then we know that's also things like gene duplication and loss, such as in metazones, can mean we have no data or more complex patterns to interpret. So here we have a gene loss, here we have a gene duplication, and here we have another duplication there as well. Recombination also obscure the evolutionary histories. And here we have recombination between the blue allele and the pink allele at this point in time, meaning that the patterns for this sample A might be different to B, C, and D, even though at the pink allele, pink locus, A and C have more recent common ancestry, compared to D, and A and B at the blue locus have more similarity compared to other samples. And that may be because there's a portion missing at this between the blue and the pink region, meaning that they're not alignable directly. And this might give rise to patterns where we see A and B be clustering more closely together at the first part of the alignment. And for the second part, we see A and C are more similar to each other. So again, this is an example where we see different phylogenetic patterns. So Turk-Kusi doing bootstrapping, trying different substitution models, and examining the tree topologies at different segments all help clarify these mechanisms of incongruence. So horizontal gene transfer, mentioned hybridization, selection can cause distortion by sampling again, can also cause distortion. We need to sample enough sites and genes. We need to infer the orthologs appropriately. We need to have the right model. We need to have an optimal amount of trimming present. And then character recoding can sometimes occur when you're looking at amino acid sequence. So there are some recombination detection methods. So there's things like LD hat and the 3-allele test, which can work for non-haploid organisms. Sequence similarity, we have things like RDP is very good at that. There's other ones like GeneConf, Simplot, Syscan, Topal, Max chi-square. We can also study phylogenetic incongruence using things like TreeSeq and other tools. We can look at this in networks, things like SplitStree and NeighborNet allow us to examine this. I can also look for convergent evolution as well, using things like homoplacy tests and other tools. So what we can do is consider phylogenies where we say we have a recombinant sample. It might appear in a tree, especially with the longer external branches, a typical signal for that. If we remove recombinants, we might see a picture like this, or we can eliminate the region, mask out the region that contains that recombinant segment. And we would do that across all the different samples, and that might mean that the external branches are relatively equal. We can also split partition the recombinant regions into a different part, or split the tree in possible. Essentially, you have two different trees reflecting the two different patterns at these different regions. We can also look at phylonests as well. Phylets incorporate reticulations from wrong combinations. They're acyclic directed graphs where every node other than the root has an in degree equal to 1 or an out degree equal to 1. So we can see this kind of pattern here where we're looking at, in this case, the hybridization of wheat, where we have different complex patterns of gene flow between the different organisms, and therefore we're looking at something more similar to this phylonet looks like a ancestral combination graph rather than a traditional phylogenetic tree. So that concludes this video on why we would get the wrong tree. Please see the practical for more information where we explore these concepts in more detail.